Hi friends, welcome you all. So in our previous lecture we have studied preservation of foods by physical method. In that we have covered thermal processing of foods, and it is carried out by process of pasteurization as well as drying as well as by means of microwave waves. And in this we have covered pretty much details of D value, Z value, then thermal dead time and other features. Then we have covered most of the details regarding pasteurization. Now in this video lecture we'll study the non-thermal processing, and this method includes treatments other than which are dependent upon temperature, and it includes different kinds of chemical method as well as radiation based methods. So the application of extreme heat treatment used for food preservation affects nutritional and organoleptic properties of food. So it is clear that after heating food, the nutritional properties of that food get altered, flavors get altered, natural aroma and other things they get altered. So in recent years, consumers demand for fresher, high quality and safe food has increased. So therefore normal non-thermal methods of food preservation for the inactivation of microorganisms and enzymes as an alternative to thermal processes are being used. So besides using thermal process, so there are some non-thermal processes. They are pretty much popular nowadays because due to by using these non-thermal processes, the nutritional properties of food, they do not get altered. However, high resistance of certain enzymes and microorganisms to non-thermal processes easily bacterial spores, especially bacterial spores, limit their application. So during non-thermal processing, the temperature of food is held below the temperature normally used in thermal processing. Therefore, minimal degradation of food quality is expected, and non-thermal processes of food preservation improves food quality and enhances safety level. So these are considered to be better over thermal processes, and overall the most non-thermal preservation techniques are highly effective in inactivating vegetative cells of bacteria as well as yeast and molds and bacterial spores. Also they get inactivated and eradicated, and most of the enzymes they also get inactivated. However, difficult to inactivate with these procedures. So thus their use is limited to food where most of the enzymatic reaction do not affect food quality, or where spore germination is inhibited by other prevailing conditions such as pH. So in overall and in general, non-thermal processes are pretty much means feasible as compared to thermal processing of food, and they are popularly and commonly used for preservation of different kinds of foods. So first, low temperature preservation. So storage at low temperatures prolong the shelf life of many foods. In our home, we keep food for in fridge, and we can consume that preserved food for 4 to 5 days, or sometimes we can consume it up to 8 to 10 days or 12 days or 15 days. Again, it depends upon food, but in home we use fridge for preservation of food, and this freezing effect is basically dependent upon low temperature preservation parameter, and storage at low temperature prolongs the shelf life of many foods. So in general, low temperature reduce the growth rates of microorganisms and slow many of physical and chemical reactions that occur in food. So what low temperature does, so it slows the growth rate of microorganism as well as it slows many physical and chemical reactions that occur in food. And low temperature are also used to preserve food by lowering microbial activity through reduction of microbial enzymatic activity. However, there are some cyclotropic bacteria are known to grow even at commercial refrigeration temperature for at 7 degrees Celsius, and these bacteria include number of genera of Pseudomonas, then alkaligenes, then micrococcus and flavobacterium. So these are the microorganisms, they still can survive at refrigeration temperature, and some of the fungi also grow at refrigeration temperature, and slow freezing and quiet freezing are used for long term preservation. So freezing reduces number of microorganisms in food, but also does not kill all of them. So in case of microorganism, cell proteins undergo denaturation due to increases in the concentration of solute in unfrozen water in foods, and damages caused by ice crystal. So basically uh, protein they get denatured by heating means keeping food at low temperature, and in this way the cells they get inactivated. So first is refrigeration. Again, this is a common method we use. So in our home also we use refrigeration, and refrigeration slows down the biological, chemical, and physical reactions that are shorten shelf life of food, and exposure of microorganisms to low temperature reduces their rates of growth and reproduction. So this principle is used in refrigeration and freezing, and microbes are not killed at refrigeration temperature for considerable period of time. But in refrigerators at 5 degrees Celsius, food remain unspoiled. But in freezer below 5 degrees Celsius, the crystals form, tear, and store microorganism. So the freezing, extreme freezing, it may kill many kinds of microbes. However, some are able to survive. So there are some species, say for example Salmonella, Streptococcus species survive after freezing, and for these types of microorganism, rapid thawing and cooking is necessary. So as far as the refrigerators are concerned, so they should be set below 12 degrees Celsius to control growth of microorganisms in food, and this lowered temperature is also reduces respiration rates of fruit as well as vegetables. So which retards the reaction that promotes spoilage. So freezer or refrigeration does not only remove the microbes or stop their microbial growth, but it also slower down the default reaction that are present in food as well as vegetables, in case of fruits and vegetables. So all perishable foods should be refrigerated as soon as possible, and preferably during transport to prevent bacteria from multiplying. So most of the time refrigeration does not kill the organism, but it stops the growth of that microorganism. Okay, and what it does, so it slows the rate of some enzymatic and chemical reactions that by default they are occur in particular type of food, say for example fruits as well as vegetables. So refrigeration is generally used to reduce spoilage during distribution of perishable foods, then increase the holding period between harvesting and processing, and extended the storage of life of commercially processed food. So all foods are not benefited from cold treatment. For example, bananas turn black, and bread goes to stall when refrigerated. So refrigeration method is not suitable for all kinds of food, but we can keep vegetables as well as fruit for longer time. And next is freezing. So it includes deep freezing as well as slow freezing. So these are different techniques of refrigeration, and these are used for preservation of different kinds of foods. So in case of deep freezing, you are known that so there are some milk derived products like desserts, then ice creams, and then cheese or butter, then all these products they are means stored in deep freezing conditions. Okay, then there are again slow freezing and rapid freezing, different types, and these are used for different kinds of foods. Okay, so preservation by freeze drying is we have already seen, we have already studied, and the next is irradiation. So generally alpha, beta, and gamma radiation particles are used for preservation of food. So these are radiations, and they causes destruction of various kinds of microorganisms, and these radiations are of high frequency with high energy content, and they have been power to break down molecules into oppositely charge units termed as ions. So how radiation affect what they do, they simply break the molecules into oppositely charged units termed as ions. So generally after radiation, ions they get formed, and these radiations are therefore called as ionizing radiations. So why we call these radiations are ionizing radiations? Because after exposure to this kind of radiation, ions they get formed, and these includes alpha radiation, beta radiation, as well as gamma radiation. Okay, so these radiations are therefore called as ionizing radiations, and the treatment has a range of effect including killing bacteria, mold, yeast, insect pest, and reducing ripening and spoiling of fruit, and other higher dose inducing sterility. So there are some doses which are designed as per the quality and type of food, and it includes killing. So effect of radiation includes killing of bacteria, then molds, then it again kills insect pest, and it reduces ripening as well as spoilage of fruit. And again, if you apply radiation for higher dose, so it eradicates complete or it completely remove the microbial flora or all kinds of microorganisms. So the technology may be compared to pasteurization. So it is sometimes also called as cold pasteurization, as the product is not heated, and eradication is useful only for foods of high quality. So this kind of technique is used for high quality foods only, and spoiled food cannot be reverted to a unspoiled state. Okay, so eradication is not effective against viruses and prions, and it cannot eliminate toxins already formed by microorganism. So basically these two are drawbacks. So enzymes they are still remain as it is, as well as these radiations they will not going to affect viruses as well as prions, and they cannot eliminate toxins. So the radiation process is unrelated to nuclear energy, but it may use radiations emitted from radioactive nuclides produced in nuclear reactors, and eradicated food does not become radioactive. So this is a good thing and main advantage, that radioactive food it does not become radioactive. So national and international expert bodies have declared food eradication as wholesome, and the food is exposed to controlled levels of ionizing radiations in the form of gamma radiation, then X-rays and electron beams to kill harmful bacteria, pests, as well as parasites, or to preserve it freshness. So these particle sources are readily available in the form of radioisotope, that is cobalt 60, and this is the most suitable gamma particle emitter. So cobalt 60 emits gamma particle, and the penetration of power of different radiation particle is different. Alpha particles are stopped by a sheet of paper, then beta particles can penetrate through 1 to 2 centimeter thick sheet, and gamma particles can penetrate even 30 to 120 centimeter thick sheet. So in case of penetrating power, gamma particles are of high penetrating power. So UV radiations are also used as an alternative means of treatment of foods, but penetration power is very low as compared to other radiation, and ultraviolet radiation is valuable for reducing surface contamination of several foods, and this short wavelength light has been used in cold storage units, meat processing plant, as well other. A rontagen R is a quantity of gamma or X-rays which produces one electrostatic unit of electricity charge of either sign in a cubic centimeter of air under standard conditions. So it is measured, these radiations they are measured in a rontagen R, and this is what it is the quantity of gamma or X-ray which produces one electrostatic unit of electric charge of either sign in an cubic centimeter of air under standard conditions. So 1 electrovolt EV is the energy gained by an electron moving through a potential difference of 1 volt, and a millivolt is 1 millivolt electrovolts. So these are specific notations for radiations. Then mechanism of microbial inactivity by radiation. So how this radiation affects microbes? So in case of radiation, radiation causes disruption of internal metabolism of cell by destruction of chemical bonds. So basically this radiation disrupt chemical bonds present in that microorganism, and after breaking that chemical bonds, the molecule present in that microorganism, they will no longer intact and they get distorted. So DNA cleavage results in loss of cells ability to reproduce. So first effect is occurs at nucleic acid, that is DNA. So once DNA get damaged, obviously that cell will not reproduce. Then free radicals are formed upon contact with water containing foods, and these free radicals react with cellular DNA causing radiation damage. So DNA is considered a radiation sensitive portion of cell. So shorter wavelength have enough energy to break knock off an electron form free radical, but not high enough to split an atom and cause target become radioactive. So interaction between free radicals and DNA is responsible for killing effect or ionizing radiation. So basically these radiations, they produces free radicals, and these free radicals when interact with DNA, the DNA structure get distorted, and once the complete DNA get inactivated, so the cell will no longer have a capacity to divide. So the ionizing radiations can cause chemical reactions and alteration of chemical in tissues, which can be toxic or fatal to humans in high dose. So much of the reactivity of ionizing radiation organism is with water and produces superoxide radicals. So basically these kinds of radiation in case of microorganism, they react with water present in that microorganism, and water is, we know, it is present in the form of cytoplasma or protoplasma, and once these radiation alters the water molecule, and it produces free superoxide radicals, and these superoxide radicals, they are toxic for that particular microbial cell. So these includes hydroxyl radicals, hydroxyl radicals and hydrogen peroxide. So these are produced during high energy collision of gamma rays and heavy metals, that is heavy elements, that is tungsten. Then there are different features of alpha particles, gamma particles as well as beta particles. So alpha particle are positively charged particle ejected spontaneously from nuclei some radioactive elements and have low penetrating power and short range. So the most energetic alpha particle will generally fail to penetrate the dead layer of cell covering the skin. Alpha particles are hazardous when alpha emitting isotope is inside the body. Then next, gamma radiation. So they are most widely used type of ionizing radiation, as they have good penetrating effect up to 20 centimeter in food depending upon exposure time, and emitted in all directions continuously. So these are mostly used gamma particles having high penetrating capacity. So these are produced by exposure of natural cobalt 59 to neutrons in a reactor, where reaction between two species produces cobalt 60 and cobalt 60. So cobalt 60 especially manufactured for radiotherapy and medical device sterilization is, and food eradication is not a waste product of nuclear receptor. Then in case of a beta particle, so it is a charged particle emitted from nucleus during radioactive decay, and it has mass equal to 1 out of 1837 then of proton, and a negatively charged beta particle is identical to an electron, and positively charged beta particle is called positron. So these are effective and the radiations kills different kinds of microorganisms, especially pathogens. In case they kills Campylobacter, Salmonella, Trichoderma, Listeria and many others, and again it delays ripening of fruit as well as vegetables, and it also eliminates insect fruit vegetables, okay, and it inhibits sprouting in onion, then potatoes, and these radiations are also replace the need of chemical fumigation. So this method is cheaper than freezing or refrigeration. So these are overall details regarding different kinds of radiation. Then next, food irradiation processes, they are of different types. So these are radioionization. So So this method minimizes pasteurization and it inhibits sprouting and delay ripening. So it kills insects and it is used for shelf life extension. So these are different kinds of food eradication techniques. So in that, first is radiorization, and here what we do in radiorization, we expose radiations to minimize it mimics like pasteurization, and it inhibits sprouting and delayed ripening. So it kills insect and it is used for shelf life extension. Then radicidation. So this method will eliminate spoilage causing microorganisms and non-spore forming pathogens, and food will not get spoiled but still may contain some pathogen. Then next, radapauperization. So this method also called as radiation sterilization, and here reduction of microorganism occur to the point of sterility. So here we can say that by using this procedure of radiation, we can eradicate completely all forms of microbial life. So here effect of radiation on microorganism includes, so we can see especially we have studied that in case of radiation they damage molecules present in the microorganism, and once this molecule they get destroyed, so the cell will no longer remain in its intact form. So how these radiation affects gram positive as well as gram negative bacteria? So gram negative bacteria are generally more sensitive than gram positive forms, and bacterial pores are strongly resistant. So yeast tend to be rather more resistant than molds, and smallest viruses they require a very few doses to achieve million-fold reduction in their numbers. So these are different kinds of microorganisms, and here respective lethal doses of radiation they are given. So in case of viruses it needs almost 10 to 40 kilograms. Okay, then yeast, whether it is fermentative or normal, it requires 4 to 9 kilograms lethal dose, and in case of a bacteria it requires 2 to 25, and these are different pathogenic microorganisms here mentioned, that is Mycobacterium tuberculosis, Staphylococcus aureus and Bacillus stereothermophilus, and these are their respective lethal doses are given in this column. Okay, so this is overall regarding radiation based food preservation technique which is used for high quality foods, and we have covered pretty much details thing about that radiation. Now pH control. So we have seen that pH affects growth of different kinds of microorganisms, and especially in case of bacteria they grow towards higher pH and up to lesser pH, and in case of fungi, so they need acidic pH for their normal growth, as well as molds even they require acidic pH condition, but in case of bacteria they requires some higher conditions of pH. So here almost every food with exception to egg whites and soda crackers has pH value less than 7. So foods can be broadly categorized on the basis of pH as high acid, medium acid and low acid. So example, so these are 4 categories on which foods are classified on the basis of their pH profile. So here high acid food includes which has pH of 3.7, and these highly acidic food includes apples, lemons and raspberries. Then acidic food they have pH range of 3.7 to 4.6, and it includes oranges, olives, tomatoes, etc. Then medium acid pH food, it includes bread, cheese as well as carrots, and it has pH range of 4.6 to 5.3. Then low acid food they includes pH over 5.3, and it includes meat, fish and most vegetable. So most of the microorganisms they can grow based in pH between 6.5 to 7.5. So it is almost at neutrality, and yeast and molds are capable of growing over much broader pH range than bacteria. So most spoilage of yeast and molds grow at pH value greater than 2. So basically the acidic pH foods they are spoiled by yeast and molds because they need acidic condition for their growth, and few of the pathogens will go below pH 4, and this information is important because it will help in determining food stability with respect to microbial spoilage. Then next, osmotic pressure is the another physical process. It is required for preservation of food, and the principle of osmosis is applied. So foods they are preserved by adding salts and sugars to them. So in our home, we every year we used to prepare pickles, and pickles are in pickles we add lots of amount of salt, and due to that salt the osmotic conditions are maintained, and we can keep these pickles for a longer period. We can keep pickles if it is maintained in good condition, so we can keep these pickles at for up to 8 months, 9 months, or for 1 year. And why these are stable? So because of high salt condition. So in high salt condition and high sugar condition, so microbes their osmotic pressure get increased, and they cannot survive. So these chemicals remove water out of microbial cells, causing them to shrink in hypotonic environment and thus stopping their metabolism. So jam, jellies, fruit syrups, honey, etc. are preserved at higher sugar concentration, and in case of fish, meat and vegetables, they are produced by preserved with salt. So this is again common practice of keeping food for longer time. So by maintaining osmotic pressure, by adding salts as well as sugars, we can keep food for longer time. So this is how physical processes are used for preservation of food. So in this lecture we have covered non-thermal processes other than thermal treatment which are used for preservation of foods. Thanks.